Hello my dear students and my friends, welcome back to another very short video tutorials on semi-conservative model of the DNA. In this video lecture, I shall be using stick notes to explain 1958 Meselson and Stahl experiment. That was a remarkable experiment to prove DNA replicate in a semi-conservative model. According to our knowledge that we know, DNA was believed to be replicated in the disruptive model and conservative model. So in this particular lecture, we will study mainly the E. coli bacteria because the original experiment in 1958 was conducted in E. coli. Further, this Taylor experiment that conducted by the Taylor et al. Conducted in the Vicia fava in plants. So first you start with the prokaryotic experiment, then it was confirmed and reproved the semi-conservative model of the DNA replication in the plants. So let's start. Here I am using two stick notes, pink color for denoting the heavy isotope N15 and N14 for the light isotope. Try to understand, here we are supposed to use the isotope of the nitrogen, that is the heavy or light, not radioactive. So what is the fundamental construction of this experiment? You need to take a group of bacteria and let them grow in the medium of ammonium chloride. So I will write down here NH4Cl, ammonium chloride. Now in this case, the nitrogen first that you have to take, that was taken in that one Meselson and Stahl experiment. Taylor was the next one. So there they use ammonium chloride, and the nitrogen was N15, heavy isotope. And when you let the bacteria grow, normally bacteria grow by binary fission, so it takes 20 minutes for the each cycle of cell division in a binary fission. So what you have to do, after a few generations, you see this nitrogen, this heavy isotope will be incorporated in the nitrogen base and let them grow in the further light isotope rich medium. So first you start with the heavy isotope, then light isotope. Now what was the result they obtained? That was very interesting. So first they took two strands of the DNA that is pink in color. So say this is the double stranded DNA, and initially the isotope was heavy, so both N15, this is the homodimer structure. Now once you let the things replicate in the light isotope, so when it split, two strands will separate DNA replication, during the DNA replication. So they found this sort of thing. But now it was not conservative. So both of the strand was not homodimer. What happened? There they found incorporation of this green paper. What is green paper? It is the light isotope. Why it is happening? Because as they are growing in the media, and media is rich in light isotope of the nitrogen, so here you get the semi-conservative model. So it is the N15 homodimer, then you get the heterodimer N15 N14. So this way in the first generation you got the result. Next you go for the second generation, so it will take 20 minutes. Again you go for the next generation. So you see terminal pink N15 remain as it was, and just you have to incorporate the green paper, that means the light isotope. So if you understand in this way, it will be easy for you to remember. Okay, let me take it. So this is the process. During the replication, these things happen. So here you can see this is the homodimer, this is the N15, pink is N15. Now this light isotope, this one, it will split, so it will stay together. So there is no heavy isotope was found. So when you go for a cesium chloride based centrifugation, you get heavy and light band like this. So you can easily prove that DNA replicate in a semi-conservative way. But not only this, we shall study also the mathematical equation, because suppose you need to continue the experiment for 3, 4, 5 generations. So for suppose you know the 2 to the power n, this is the formula we use in every 20 minutes. So n is 1, means there will be 2 set after first generation. If you see 2 to the power 2 in the second generation, you see 1, 2, 3, 4. This sort of DNA you get. So this is your N15, N14 heterodimer, this is homodimer and heterodimer. So there is a equation for it, 2 to the power n minus 2. If you take this This concept, this is the number of heterodimer that you are going to obtain. For example, you are running the experiment for 3 generation. So it is a 2 to the power 8. You know, only 2 will be the heterodimer and this number will be homodimer. Okay. What I am trying to say, suppose you run it for the 4th generation. So number of DNA will be obtaining 2 to the power 4. So that is 16. So out of 16, try to understand 2. This is the number. This number is always constant because one strand, this pink paper is always fixed. So you get only 2 as a heterodimer. Correct? You correct the concept. So this is your heterodimer. And remaining whatever the number of cycle it goes in every 20 minutes, so rest of the things will be homodimer. So after 4 generation, you see 16 is the total heterohomodimer set that you can separate in the cesium chloride based centrifugation. The 14 will be the number of homodimer. That means here you see 50%, so you get the percentage of it, right? In the first case, you see there is a no homodimer formation because both of them are hetero. But here again, this will again, because the medium is N14 medium, so it will definitely incorporate the light chain. This proves that it always go in a semi-conservative way when one parental strand is always conserved. So it is a semi-conserved. Semi means half of the parental strand, that is anti-parallel to each other. So this is, say this is this one and this is conserved here. So in every step of the generation, you see semi-conservative replication, semi-conservative DNA replication. And when you go for the tail, I think you can see the picture. Look at the picture carefully. Now, you know, in the Taylor experiment, it was not ammonium chloride, it was a radioactive isotope that was used by the Taylor, correct? I hope you understand the things. Now, if it is done, then come to the concept of the VCFABA. In the Taylor experiment, he used the thymidine, that is a radioactive isotope. So, same way you can represent the thing because DNA contain many important molecules excluding sulfur. You see it contain nitrogen, it contain hydrogen. Definitely, so by using different tracer technique, radioisotope technique, you can prove it. But this is not a radioactive process. So in this way, you can calculate the number of homodimer and heterodimer in n number of generation with a very easy manner, and percentage and the ratio also you can take the percentage of homodimer. Say here homodimer is 14, heterodimer is 2, so it is always 2 is to 14, so 1 is to 7. In that way you can proceed. I hope this video will help you to understand 1958 very popular Meselson and Stahl experiment. Thank you so much for watching.